Our next presenter is Dr. Jing Wang, who is from the Fruit Tree Research Institute of the Guangdong Academy of Agriculture Science. She is an associate researcher, and she graduated with a PhD from the Department of Biology at the University of Munich, Germany. Currently, her work is dedicated to the innovation utilization of Longan germplasm resources and research on green, high yield, and high quality cultivation techniques. By combining single molecule sequencing technology and chromosome conformation capture technology, along with colleagues, she successfully deciphered the Longan gene sequences and obtained the gene sequence and obtained the first chromosome-level Longan reference genome, which is of great value for the research of Longan's functional genome and molecular breeding. Currently, more than 10 papers have been published in journals such as Horticultural Research, Journal of Experimental Botany, Plant Physiology, and BMC Plant Biology. So we're going to some heavy stuff now. It's a lot of molecular explanations here. So Dr. Jing Wang will present on investigating the mechanisms of unilateral cross-incompatibility in Longan. Dr. Jing Wang, please proceed.
Hey everyone, good morning. I'm Jing Wang from China. Thanks to TFNet for organizing this webinar series of tropical fruits on Longan. It's such a great honor to be here with you all. Today, I want to talk about my research work on Longan, especially the topic of investigating the mechanisms of unilateral cross-incompatibility in Longan. UCR is short for unilateral cross-incompatibility. This is my email address if you need. But first, let me give you a quick introduction of myself. I got into fruit tree science at Shandong Agricultural University and received my bachelor's degree there. And then I did master's at Northwest Agriculture and Forestry University and majored in biotechnology of fruit trees. After that, I went abroad to study in University of Munich in Germany, and I worked with Prof. Daria Lester. He is a big expert on photosynthesis and chloroplast. When I returned to China, I spent 2 years as a postdoctor in Shanghai Normal University, still doing research on chloroplast. Since 2018, I've started working in Institute of Fruit Tree Research in Guangdong Academy of Agricultural Science. Our institute is in Guangzhou City, Guangdong Province, which has the largest Longan planting area in China, nearly 126,000 hectares. There is a Longan germplasm resource nursery here. This is a short video of the Longan resource nursery. It was founded in 1978. It covers an area of around 2 hectares and currently saves more than 150 Longan germplasms. So, okay, let's come to the contents. It was divided into 4 parts. Part 1 is Longan genome and population genetic diversity. Part 2 is boron deficiency contributes to Longan UCR. Okay, part 1, Longan genome. In 2023, we issued a first high-quality chromosome level Longan genomes. Its size was 455.5 MB, and nearly 99% of sequences, were anchored onto 15 chromosomes. In order to exploit the genetic diversity of Longan germplasms around the world, including southern part of China and some South Asian countries, we performed deep genome sequencing of 87 Longan accessions, most from Guangdong and Fujian province in China, the rest from Thailand, Vietnam, and Australia. Based on SMP data, the Longan germplasms can be clustered according to their distributions. It means that Longan biogeography is a contributing factor for genetic diversity of Longan. Also, the maximum likelihood trace shows that Thailand populations were distant to Chinese populations. It's well known that Yiduo is the main cultivar in Thailand with a very good taste of fruit. And Shixia is a main cultivar in China with a good fruit taste, but smaller fruit size. So we expect that if we cross Yiduo with Shixia, the heterosis will exist with super good fruit quality. Before I give you the result of this crossing, I would like to show you that we have some contact with major university in Thailand. This is the vice president, Professor Pavan. He is called Mr. Longan in Thailand. This is Professor Tilanu. Both two persons are very friendly. This international contact is very good for my further research. Since 2018, I did crossing between Yiduo and Shixia every year. Unfortunately, unilateral cross-incompatibility happened when using Yiduo as the female parent and Shixia as a male parent. We speculated whether this was due to boron deficiency, because the soil in Guangdong orchards had a boron content of only 0.17 milligrams to kilograms, while in Thailand it can reach up to 5 milligrams to kilograms. To investigate this further, we conducted a boron application experiment. When successfully restored the cross-compatibility between Yiduo and Shixia, allowing Yiduo to bear fruit. This confirmed that boron deficiency contributed to unilateral cross-incompatibility in Yiduo. In order to understand the underlying reasons for boron deficiency in Yiduo and how to increase its levels, we compared the phenotypes of Yiduo and Shixia, including leaf phenotype, microscopic observations of leaf transactions, and detection of boron levels in different organs such as roots, stem, leaf, and flowers. Interestingly, Yiduo was a high-demanding cultivar, despite the boron levels in its leaves and flowers higher than Shixia cultivar. At molecular level, boron transporter play important roles in boron levels in plants. So we did homologous strain family analysis of BOR transporter and NRP transporter. Overall, there are 5 BORs and 33 NRPs. And then we compare the expression levels of these genes at different periods with various plant organs. As a result, it shows that the NRP19 was expressed only in vegetative organs of Yiduo, while NRP1 was exclusively expressed in the flower of Shixia. Further functional characterization of NIP90 and NIP1 revealed that both two were localized on the cell membrane as expected. And we obtained NIP90 and NIP1 overexpressed pollens respectively by the method of pollen magnetofaction, and cultured them on the medium supplemented with different concentrations of boron. According to the pollen tube growth rate, NIP1 preferred high boron conditions, indicating its role as a potential importer for female flowers, whereas NIP9 functioned primarily as an exporter for vegetative organs. Also, we utilized NIP1 or expressed pollens to pollinate Yiduo. It can rescue the cross-compatibility of Yiduo. Additionally, we conducted a screening of Longan germplasms with higher boron levels as potential rootstocks, as Longan seedlings in China are typically propagated by grafting. One particular Longan accession, called HLGY, contained nearly 300 milligrams to kilograms of boron in its organs. In the future, we plan to utilize HLGY as the rootstock for Yiduo. During the boron application experiment, we observed that there is a downregulation of yasmonic acid level in the female flower of Yiduo. This led us to investigate whether GA could influence UCR in Yiduo. Microscopic observations of pollen tube growth in oil and comparisons of pollen tube growth rate show that when Yiduo as a female parent, it inhibited pollen tube growth. Measurements revealed that both GA and its active form GARLE were consistently higher in content with Yiduo compared to Shixia. To test our hypothesis about GA's function in UCR, we prevented the biosynthesis of GA using chemicals as ibuprofen, which successfully broke the UCR phenomenon observed in Yiduo. This confirmed that GA can indeed regulate UCR in Yiduo. Through transcriptomic analysis, SPH5 is a self-incompatibility protein homologue was identified with significantly higher expression levels in Yiduo than in Shixia. Its expression levels were regulated by GA. According to sequence analysis, SPH5 is a small peptide with only 138 amino acids, capable of being secreted into extracellular space. In Arabidopsis, SPH5 overexpressed lines inhibit pollen tube growth, similar to UCR phenomenon. Further experiments, including yeast-to hybrid assays, demonstrated that SPH5 can interact with pollen-expressed membrane protein kinase MRK1. So we attempt to obtain transgenic pollens through magnetofaction mentioned earlier in order to clear the exact function of SPH5 and MRK1. And then we use the transgenic pollens for pollination, resulting ultimately transgenic seedlings. Until now, we already got MRK1 overexpressed pollens and SPH5 overexpress Longan seedlings. Because it's still challenging to easily and quickly create genetically modified Longan plants with tissue culture. So as we know that it's not easy to get transgenetic lines of Longan, here we suggest you can to do some pollen magnetofactions. It can work. Perhaps next year I would present you some interesting results of this project. In summary, SPH5 functions in the downstream of GA-regulated UCR. SPH5 was secreted into extracellular space to interact with pollen-expressed kinase as female pollen interaction. However, there are still another two questions to discuss. Exogenous boron can facilitate fruit settings of Longan indoor. Is there other environmental factors necessary? GA biosynthesis inhibitor can also block UCR by decreasing the expression of SPH5. So how does GA regulate SPH5? Then the part 4 is the additional project on Longan. It's well known that Longan trees can grow higher and higher as time going on. So its management is time-consuming and labor-intensive. So we are exploring mechanized cultivation mode of Longan. And we mainly focus on tool management, including mechanized pruning mode and some new techniques of fruit detection. In terms of mechanized pruning, we have explored 3 kinds of moves, including topping the head, hedging one side, and also the former 2 together. Additionally, for fruit detection, we are utilizing FT-NRR analyzer. It relies on near-infrared spectroscopy. Alongside this method, we are using exact cameras to detect the phenotype of the fruit. Hopefully, we can find some correlations between them. Finally, I would like to express my thanks to my colleagues. Professor Li is our team leader, as well as Professor Han and Professor Hu. Also, I would thanks to Dr. Huang, Liu, and Wei. Furthermore, I'm grateful for collaboration with Professor Li Guo from School of Advanced Agricultural Science, along with my assistants Yun Wang and Boyun Wang. Special thanks to both fundings from Guangdong province, as well as my thanks to you.
Okay, thank you. Thank you very much, Dr. Jing Wang, for that very technical presentation. I think you have 4 parts. The first is on the genome sequence. Second one is on the effect of boron. And then you spoke about using the pollen, pollen to get into, well, to get transgenics, to get the transgenics planting material. And the last one is on mechanized pruning. So thank you so much. Maybe there'll be among us who are very heavily technical who wants to ask Dr. Jing Wang questions. You can wait till the end, but I'm sure there'll be some who are interested to ask you some questions. Thank you very much for sharing with us your research and also what you do, to improve the Longan, in China, basically. Yes, thank you so much.